Good afternoon, everybody. Thank you for joining. My name is Vip Jairath. I'm a gastroenterologist and Professor of Medicine at Western and CMO here at Alimentiv. I'd like to welcome you to our fourth live stream event. We've had a number of these now, and the last couple of events on precision medicine and IBD, and then intestinal fibrosis and Crohn's disease, really integrated basic science, clinical medicine, and drug development pathways. Our topic today is implementation of histopathology in gastrointestinal clinical trials. I'm really pleased to see that over 600 people have taken time out of your busy schedules to join us today, with people from academia, industry, and the fantastic panel of speakers that we have today. I think we're really fortunate to be joined by some of the leading experts in this field, and their vision has really helped move this area forwards. There's going to be 4 short talks today, and we're going to cover both NASH and inflammatory bowel disease. And then there'll be a clinically focused talk followed by a histopathology focused talk in each of those. Just in terms of some housekeeping, after the keynote presentations, there'll be a panel discussion. Your lines are muted. You can feel free to submit questions in your chat box through the presentation. We'll endeavour to try and answer some of these during and after the presentations. And if we can't get to them due to time restrictions, we will try and get back to you by email. So we've got a pretty full agenda, so I'd like to get started. So we're going to actually start in the liver rather than the gut, and I'm really pleased to introduce our first speaker, Dr. Stephen Harrison. Dr. Harrison earned his medical degree from the University of Mississippi School of Medicine. He did his internal medicine and residency and GI fellowships at Brooke Army Medical Center and advanced liver fellowships in St. Louis University. He's board certified in both GI and internal medicine. He's a visiting professor of hepatology at the Radcliffe Department of Medicine in Oxford. And he's really internationally known for clinical studies and trials in Hep C and non-alcoholic fatty liver disease. He's extensively published in these areas and really on multiple phase 3 clinical trials and drug development programs. Dr. Harrison's the medical director for Pinnacle Clinical Research and the president of Summit Clinical Research. And he's going to talk to us today about histopathology as a treatment target in NASH clinical trials. Thank you, and we look forward to your talk. Thank you, Stephen.
Well, thank you very much, Vip, for that wonderful introduction. And it's an honor and a privilege to be here today to talk to you guys about something near and dear to my heart, fatty liver disease. So this has been really an evolution over the past 10 or 15 years as we begin to drill down on our first FDA-approved treatment for NASH. And it's been a large time in the making, but I want to focus in on histopathology as a treatment target and how we started out and how we have arrived to where we are today. So this is my agenda. We'll just talk about an overview of the evolution of histopathology for enrollment in trials. Then we'll review associations between histopath and longer-term clinically meaningful outcomes, which is really the hallmark of what the FDA is looking for with drug trials. And then we'll review some recent drug programs, as well as results and effect sizes for histopathology. So, evolution of histologic endpoints really begins with understanding the natural history of this disease. For most people with fatty liver, they grow old with their disease. With about 25% of them, however, there's risk of developing non-alcoholic steatohepatitis, progression through fibrosis to a cirrhotic endpoint. Now, we know that cirrhosis is most closely linked to liver-related mortality. However, NASH with fibrosis, this what we call at-risk NASH population, is also at risk for increased morbidity and mortality. So as we think about where we are today, the current FDA approvable endpoint for conditional approval of your drug would be histopathology based, and that is either NASH resolution without worsening of liver fibrosis or fibrosis improvement of at least one stage without worsening of NASH. Or you could have both. Now, that just gets you conditional approval. For full approval, you have to do a longer term outcome trial. So usually we keep those people enrolled in the phase 3 trial after reaching Subpart H approval to look for progression to cirrhosis or a clinical endpoint that you see listed below. So how did we get here? Well, the first trials, the first large trials in NASH predated the FDA approvable Subpart H endpoint. And this was the Pivens trial and the Flint trial. And here, the inclusion criteria were very pragmatic: definite or possible NASH, NAS or NAFLD activity score of 5 or more, or definitive NASH with a NAFLD activity score of 4 or more with at least 1 point in ballooning. And that primary outcome measure is really just improvement of ballooning by at least 1 point or more, no increase in fibrosis, and either a decrease in the NAS to a score of 3 or less, or a decrease in the NAS of at least 2 points. So it's very complicated. And so we've tried to evolve that a little bit. The Flint trial in 2011 looked at really a decrease in the NAFLD activity score of at least 2 points without worsening of fibrosis. And this has subsequently become one of the endpoints we look at in, say, an earlier phase trial, like a phase 2B trial, where we're just looking to see if we're moving the needle in the right direction relative to a NASH phenotype. Now, just shifting gears to where we are today. So again, the definition of resolution of NASH from the Golden 505 trial, which was in phase 2B trial with elafibranor that predated the Resolve It phase 3 trial, that initial disappearance ballooning or inflammation. And that was subsequently modified at the end of the trial to apply the new modified regulatory definition, which is the definition of NASH resolution being absolutely no ballooning and inflammation scores of 0 or 1. So that's currently where we reside today: ballooning of 0, inflammation or 0 or 1, with really no deference given to steatosis. So where are we moving towards? If that's where we are today, where we're beginning to see is the advent of fully quantitative assessment of each of the component parts of the NAFLD activity score, as well as fibrosis. And in this slide, we see from data from the Stellar and the Atlas studies. On the left is placebo in blue, and then a combination of two drugs, selifexor, which is an FXR agonist, and farsocostat, which is a acetyl-CoA carboxylase inhibitor. When put together, we see on the y-axis the response rate at week 48. And you see that there is a steady improvement. However, if you look to the right, the way right, we can overlay this with fully quantitative assessment of collagen, and we can get at a better quantifiable amount of fibrosis improvement as well as NASH resolution. So I think the shift here is beginning to occur. We're still doing semi-quantitative assessment using those Subpart H criteria I've already mentioned. But now the future is we're going to begin to overlay this fully quantitative assessment on top to initially help the pathologist in their interpretation, with the idea that we'll improve our inter and intra-observer variability statistics, but eventually, hopefully, allowing for a transition to a more fully quantitative assessment of collagen. But I think that right now, the two go together and really augment and synergize together, fully quantitative and semi-quantitative assessment. So where are we at with our current trials? So this is a listing of the NASH histologic endpoints in the current phase 3 trials. Quickly, the elafibranor trial is with, or the GenFit trial, the Resolve It trial is with a drug called elafibranor. And there, the histological endpoint for phase 3 was resolution of NASH without worsening of fibrosis. They did not meet that endpoint. Now, with Intercept and the Regenerate trial with obeticholic acid, it was also resolution of NASH without worsening of fibrosis. There was also an alternative primary endpoint, which was fibrosis improvement without worsening of NASH. They did not hit the first one, resolution of NASH without worsening of fibrosis, but they did hit the second one, which was improvement of fibrosis without worsening of NASH. As you know, they submitted their NDA, and the FDA gave them a complete response letter. So they're working through resubmitting that for approval now with additional data. Ongoing, we have the Madrigal Maestro NASH trial with a drug called resmetirom, a thyroid hormone receptor beta agonist. The endpoint there is resolution of NASH with a little twist. It has to be at least a 2-point reduction in the NAFLD activity score without worsening of fibrosis. The GalMed trial is with a drug called Aramcol. It's an SCD1 inhibitor. It's a dual endpoint, resolution of NASH without worsening of fibrosis and fibrosis improvement of at least 1 point without worsening of NASH. That trial is currently on hold. They're doing some additional work before opening that back up again. The last two studies were actually halted. The Sinacrivaroc trial is with a drug that was looking at fibrosis improvement without worsening of NASH, and that study has stopped. And the selonsertib trial, which I helped to lead, had an endpoint of fibrosis improvement without worsening of NASH, and that was a negative study. So we've had a bit of a challenge getting across the finish line with phase 3, but I'd say the future looks bright, and we've taken our lessons learned and are really using those lessons to help improve the way we enroll these future trials. So just shifting gears to talk about some important data linking histopathology to outcomes. So we know fibrosis portends a worse prognosis, and this slide really drives home that point. You can see in the red line in this Kaplan-Meier curve that cirrhosis actually is linked to a worse clinical outcome. And we know that stage 3 is also linked to a negative outcome, as is stage 2. And that's why the FDA is really mandating including at-risk NASH as that with F2 or greater fibrosis. Now, this shows data from the Golden 505 trial with elafibranor. And I just want to bring your focus to the left bar graphs. So in dark blue is worsening, and on the y-axis is percent patients. So on the horizontal axis is change in activity with ballooning and inflammation. So as you improve ballooning and inflammation, that's where we see the fibrosis improvers. Look to the hard left there. A -3 means you've improved ballooning or inflammation by at least 3 points. And we see almost 60% of the patients having fibrosis improvement. Alternatively, way to the right, if you worsen ballooning or inflammation by at least 2 points, that's where we see a large majority of patients worsening their fibrosis. And more recently, some data presented by a good friend of mine, Arun Sanyal from Virginia Commonwealth University, looking at data from the STELLAR-4 and simtuzumab trials, focusing in on the cirrhotic cohorts in these trials. What we see is that if you're able to improve fibrosis, that's where we see improvement in liver-related events. This is very, very important because what this says is you can use fibrosis improvement to actually predict the probability that you'll have less events downstream. And I think we're going to use this data in a very provocative way moving forward. So again, more data to support this notion. This is work by Dave Kleiner published with his colleagues in JAMA in 2019 from about 400 patients from the NASH CRN studied that showed fibrosis improvement actually with improvement in disease activity. So as you go from NASH to NAFLD, you actually see this improvement in fibrosis, and that's illustrated on the left-hand set of bar graphs. As you go from NASH to NAFLD, you see the development of more of a percentage of stage 0 fibrosis. That's the dark blue. Alternatively, as you worsen disease, fibrosis worsens as well. And that's on the right-hand set of bar graphs, the first 2 bars. As you go from NAFLD to NASH, you don't see any orange in the first bar. In the second one, you see about 18% that are F3. So you actually are worsening fibrosis as you worsen disease activity from the Golden 505 trial. And here's some data from Elizabeth Brunt, a world-renowned pathologist from Washington University and St. Louis University, published in 2019, some data looking at the Pivens trial and the Flint trial, showing the exact same thing. As you go from NASH to NAFLD, we actually have more fibrosis improvement. And the take-home message here was that the histologic features associated with fibrosis improvement were NASH resolution, improvement in the NAFLD activity score, and ballooning degeneration primarily. So finally, just a couple little slides to illustrate where we are in the disease landscape. This is fibrosis improvement, looking across the waterfront, going from left to right as you increase the trial duration from 24 weeks to 78 weeks. You see a wide range of fibrosis improvement. You also see a wide range in the placebo response rate. And I would just highlight for instance, with obeticholic acid, the drug that did meet its FDA endpoint of fibrosis improvement, it's 23% versus 12% for placebo. So a placebo-adjusted response rate of around 11%. So that kind of gives us a foundation from which to build moving forward. And you see drugs in development like efruxifermin, which is an FGF21 analog, actually showing about 62% fibrosis improvement versus none for placebo in as short as 12 weeks. Now, granted, in this particular study, only people that had an MRI improvement actually underwent biopsy. So only 2 patients in the placebo group met that. So the numbers are very small, so take that with a grain of salt. The take-home message is that we're improving our studies, we're improving our outcome measures, so the future looks very bright here. NASH resolution, very similar landscape, where we see rapid improvement in our ability to resolve NASH, but still we're troubled by the placebo response rate that appears to be widely varied. And so we're looking at ways to improve that. And one of those ways might be that overlay of artificial intelligence that I've mentioned earlier. And finally, both NASH resolution and fibrosis improvement. And you see the different studies that have looked at this, ranging from, around 19% all the way up to 37%, placebo-adjusted responses of around 20 to 22%. So the future does look bright at hitting both of these endpoints. So with that, I think I will pause, turn it back over to Vip, and thank you for the opportunity to speak today.
Stephen, thank you very much for that wonderful overview. And then it was a big topic to do in a short period of time. And I just have time for a couple of questions on the liver talk before we move on. So I'd like to introduce our next speaker, Dr. Mark Valasek. Mark is a physician scientist. He's a practicing GI and liver pathologist. He focuses mainly on innovations in diagnostic applications. He participates in numerous clinical trials related to GI diseases, and he's a clinical faculty member at the University of California, San Diego, where he's associate professor of pathology. His particular interest in practical molecular imaging and digital pathology techniques, including AI, for the evaluation of GI diseases and emerging technologies. He was awarded the San Diego Medical Society's 2014 Top Doctors, Physicians of Exceptional Excellence in the field of pathology. And he also established the first GI Pathology Fellowship at UCSD and the first director of the GI pathology section. Mark will speak to us today about measuring histopathology in NASH trials. So thank you very much, Mark, for speaking, and over to you.
I'm excited to talk to you about histopathology in NASH clinical trials. This is a basic talk, but I hope that some of the concepts will spur discussion and lively questions regarding this topic. This is our beloved balloon cells that all of your pathologists love. We have enlarged cells relative to their peers adjacent to them, and lacy reticulated cytoplasm. This is a slam dunk. All pathologists can agree on these as being balloon cells. Just a little bit of background information on NASH that we all really know. NASH was separated out from alcoholic liver disease in 1980, and over the past 40 years, we've only seen growing importance of this disease, and we now have fatty liver disease as the primary #1 indication for hepatic transplantation. On clinical biopsies, we obviously do these cases to exclude other diseases, distinguish between steatosis and steatohepatitis, and to give a fibrosis stage. And some of the newer terminology is referring to NAFLD and NASH as metabolic associated. So we may all be converting to MAFLD and MASH. And we know now that genetics are very important in this disease, including susceptibility factors and also some protective genes as well, including the hydroxy steroid dehydrogenase gene. We all know that NASH histopathology is important in clinical trials. As Dr. Harrison alluded to, patients must meet inclusion criteria, and the pathologist needs to exclude other disease entities during enrollment phase. And we talked previously about these endpoints, but some of the basic endpoints are resolution of NASH without worsening of fibrosis and improvement of fibrosis stage greater than or equal to 1. The NAS and NASH CRN fibrosis stage have been used in the vast majority of trials, and reading paradigms are not well standardized. There can be inconsistent pathology reads, and that may be one of the issues in the field. And we're also concurrently seeing emergence of AI techniques, imaging modalities, and clinical scoring systems. So there have been numerous studies showing variable agreement scores on these clinical trials endpoints. Won't go through a comprehensive review, but this is an example here. And you can see on the right-hand column an unweighted kappa in the range of 0.3 to 0.4, which is fair agreement. So this is something that definitely improved upon. Many studies have been done to try to improve agreement among pathologists. One example is the FLIP algorithm applied to the SAF score. And we don't need to get into the details, but the agreement after a training session, and that applies even to general pathologists, not just expert GI and liver pathologists. So because of this, we decided to embark on standardizing interpretation of liver biopsies. And so we convened an expert panel of 14 liver pathologists and looked at 130 items and deemed them as inappropriate, uncertain, or appropriate, and voted to compare different scoring systems. And what we found was not too much of a surprise, is that there's not universal agreement regarding the best scoring systems to use. And alternative methods were needed to evaluate ballooning and fibrosis. And then steatosis was also considered of uncertain value, as Dr. Harrison alluded to. Current staging systems do not capture the full spectrum of fibrosis, so we need extended measures to look at fibrosis and also evaluate fibrosis regression. To extend on this work, the NASH CRN, Dr. Kleiner and colleagues looked at histologic features that are prognostic. So as you know, the NAS was developed prior to some of the clinical trials and was developed as to characterize the disease state and is not necessarily prognostic. So they looked at factors that were associated with progression of fibrosis, and strong features were ballooning, portal inflammation, and Mallory-Dink bodies. And limited associations were steatosis, lobular inflammation, and total NAS score. And also there were some clinical features associated with spontaneous regression, including a low AST. So this did not encompass all the histologic features we see in these cases, and a novel histologic feature that was evaluated pericentral ductular reaction. Now, normally we think of ductular reaction as a portal process, but they were able to identify centrilobular centrilobular ductular reaction that indicated injury at that site and was associated with fibrosis. And this is actually a novel approach because they used glutamine synthetase to show that these areas were not periportal, rather they were centrilobular. So glutamine synthetase plus CK7 was used to identify centrilobular ductular reaction. And you can see on this table on the right that there was a significant association with progression of fibrosis. So there are novel histologic markers still out there that may not be included in current grading and staging systems. So to expand on this, we looked at inter and intra-rater reliability of scoring in NAFLD histologic indices. And we looked at 40 existing histologic slides of NAFLD patients. 4 pathologists read these out in 2 sessions, greater than 2 weeks apart with a washout period. And we looked at Brunt criteria, other scoring systems listed above, and then additional items including Mallory-Dink, portal inflammation measures, and expanded measures of fibrosis. And then a visual analog score was also given for the overall impression of fibrosis and other features. So we utilized the Alimentive Central Image Management System, and all indices were scored. And some of the results are what you see here. For correlation of fibrosis, so the VAS is basically your pathologist overall, the stage of fibrosis. Steatosis did not correlate with the overall impression of fibrosis. Additional measures that correlated well with fibrosis were the expanded NASH CRN fibrosis. So this is substages for bridging fibrosis, 3A, 3B, and 3C. And then you can see other measures here on the right-hand column that correlated. We had very good inter-rater and intra-rater ICCs on the expanded ballooning score and the expanded fibrosis scores, which is encouraging. And one of the important things I think that we learned from that study is that you can get good agreement of an expanded fibrosis score to try to get at if there's any regression. So anything we can do to increase the granularity is probably a good thing. Histopathologic features of regression are specifically defined in the literature, and this may be something that we can start documenting on our cases as well. So this is obviously extremely important in any study, but especially studies directed at anti-fibrogenic or fibrolytic studies. Some of the features of regression include attenuated bridging fibrosis, a pulled taffy appearance, perforated or interrupted bridges, hepatocytes with septal or portal within septal portal areas, and isolated but small, dense collagenous foci. So I apologize for that. So on this, can you guys see this? So on this slide, you can see in figure B, you can see that there's an interrupted bridge. It's also present in C. And then in D, you can see very small isolated foci of collagen that happens to be thick but very small. So these are all evidences that fibrosis has regressed in this patient. So specific documentation of that may be helpful. A brief word about adjudication. This has been a hot topic in pathology to try to get more consistent reads on histopathology. The 2+1 model is one option, where adjudication is performed by a third pathologist if there's disagreement. In this case, we would need to precisely define what constitutes a disagreement. That could be any disagreement on any feature, or other thresholds could be applied. And this would be similar to what is being done in some of the IBD endoscopic reads. Another option is having two central reads that are summed or averaged with no adjudication of disagreements. One reason I like this method is that, for example, if you have a stage 1 and rather a grade 1 and a grade 2 steatosis, for example, and the actual amount of steatosis is 30%, then potentially your one pathologist may grade it as, graded as a 1, and one may grade it as a 2. And so the average or the sum may actually be closer to the real amount. So in some ways, that's good to keep the independent data and then also the final data. Another option is to do a panel, and this is done in the NASH CRN. It simplifies the statistics. There's potentially faster processing of cases, but what if unanimous agreement cannot be obtained on histopathology? And will seniority or personalities impact the direction of the consensus? Those are some potential questions. As an adjunct to all these types of adjudication procedures, direct comparison of screening and follow-up biopsies is also a possibility. Pathologists could be blinded to the treatment group and time point, and then directly compare. And I think this is a big opportunity, as alluded to earlier, for AI algorithms to help quantitate direct comparisons between screening biopsies and follow-up biopsies. So just a final word about AI-based image analysis. There are many academic and private groups working on NASH image analysis. And one example was recently published in February as a collaboration between Gilead and I think this was with PathAI, correct? They used a machine learning model called Delta Score that was mentioned earlier and found good concordance with pathologists for steatosis and fibrosis, but lower concordance in the row scores for hepatocellular ballooning and lobular inflammation. A liver fibrosis score was calculated as a continuous variable, which is a definite benefit, and ratios of histologic features were easily calculated, which is also a nice feature. So overall, what are our opportunity zones in histopathology? There are many. We need better standardization of feature documentation. We need better standardization of central reading. We also need to, I think, augment histopathology with biomarkers. And as you know, these can come from clinical and laboratory values, from radiological imaging, from AI-based features that are either novel or standardized. and ancillary studies, including IHC. And I think also we need to establish clear methods on how to evaluate fibrosis regression. And so there are all other features that we need to do, including standardized histopathologic staining of standard stains. And as always, another issue in pathology is considering sampling as well. So these are all issues that we can discuss, and I hope that we get some lively questions regarding these. So thank you for your time, and I appreciate it. Back to you, Vip.
Okay, great. Thank you so much, Stephen and Mark, for those two talks. We're going to just take 2 or 3 minutes now to go through a couple of questions, and then we'll move on to the IBD section. So, maybe I could start. Dr. Harrison, I was quite struck by the 2 or 3 slides you had of efficacy versus placebo rates in those trials. And, I'm going to do what we're not supposed to do, which is just to eyeball and compare. But I was struck by the variation in placebo rates. And can you give a bit of insight from, your experience of why there's so much variation? Does it mean that you need another modality to qualify patients to control that a bit? Because being unpredictable is a bit of a problem for development programs. Could you give us your insights on that, please?
Placebo response rates are really an area of intense investigation, trying to normalize these across the different studies. And it really drove home when we saw the elafibranor data from phase 3. The response rate to elafibranor on fibrosis was the exact same as it was for obeticholic acid 25 milligram. But yet the placebo response rate was significantly higher than what was seen in the obeticholic acid program, so much so that the difference between the drug and placebo response was null, was negative. So it was a negative study. But in obeticholic acid, the placebo response rate was 11%. And so that delta actually led to a positive p-value. So there are a lot of speculation as to why the placebo response rate is so high. I think it's multifactorial. And I think one of the biggest drivers of placebo response rate is we don't do a very good job of quantifying and measuring lifestyle modification that happens across a very long phase 2B or phase 3 trial. We tell people very pragmatically to lose weight and exercise and diet, but we don't track how well they do that. And I think that's an area of intense focus moving forward, is how do we better quantify, for instance, their cholesterol intake. We know cholesterol, particularly oxidized phospholipids, drive underlying inflammatory processes and fibrosis. So if we're able to quantify that better, and we have apps that are being developed to help us do that, I think we'll get at better stabilization of placebo response. And then additionally, where we can take that digital AI and superimpose it on a semi-quantitative read and help our pathologists adjudicate these liver biopsies, I think that's also going to stabilize not only the placebo response, but give us a much more stable drug response rate as well. So there's a lot of work to be done in the area, but we're at least understanding what could be causing this disparity in placebo response. Now we have to try to improve on it.
Great. Thank you so much, Stephen. And maybe I could keep with another question there. So, I mean, of course, in an ideal world, nobody would have a biopsy. I don't think any patient would volunteer if there was a non-invasive methodology. Do you see how far do you see this down the line where the liver field, the NASH field, particularly in trials, may be at a stage where imaging with biomarkers could replace biopsy, both going in and as an outcome measure? Do you see that as a realistic target, and how far away?
I do, but let me be a little provocative with you here. I think we're going to separate this out into a well-comp cirrhotic population and an F2, F3 population. And I think drugs that are really focused on F4s are going to much more quickly adapt the non-invasive motif. And I think MR elastography has the ability to really drive that forward. We know now from recent data published by Alina Allen's group, as well as Mazen Nareedin's group, that MRE can actually correlate with outcome measures. And so imagine a scenario where we're doing a well-COMP-F4 trial with liver biopsy, but we're also getting MR elastography. Let's say it's a large phase 2B, and we're able to correlate improvement in histopathology with improvement in MR elastography. We're able to quantify that degree of improvement in MR elastography. I think that opens the door for a phase 3 trial in non-invasive technology, as MRE is the primary endpoint. As the slide I showed you from Arun Sanyal points out, if we're able to improve histology, that's linked to a better outcome. Now all we need is one more piece of data to confirm the link of improvement in histology with improvement in MR elastography. And I think that allows us to take the next step forward. Similar things will happen in the non-cirrhotic population trials, the F2-3 studies. It's just going to take us a bit more time to get there, but we do have some very nice biomarkers that are in development. I don't think they're quite as focused and as far along as MR elastography is, say, for a well-COMP-F4 patient. But the future looks bright for both.
Great. Super, Stephen. Thank you so much to both you and Mark. I think in the interest of time, we're going to move on and move out of the liver now into an area where I'm more comfortable, which is the gut. I just want to quickly mention, I know there's been a couple of screen freezes on the other end, and apologies for that, but the recording link will go to everybody afterwards. So I'd like to now move on and introduce our next speaker. So I'd like to introduce Dr. David Rubin. I know in the field of IBD, David doesn't need much introduction, but by way of background, David is the Joseph Kirstner Professor of Medicine. He's Chief of Section of GI, Hepatology and Nutrition, and he's co-director of Digestive Disease Center at the University of Chicago Medicine, and his medical degree from the University of Chicago, and he did his residency in internal medicine training there, as GI training as well, and medical ethics. David's a fellow of multiple professional societies, is the chair-elect of the Scientific Advisory Committee of the Crohn's and Colitis Foundation. He's had numerous awards for clinical research over the last 15 years. And last year, many of you will have seen David receive the Sherman Prize for Excellence in Crohn's and Colitis. He's Associate Editor for Gastroenterology, and some of his current research interests include biocenter monitoring in IBD, prevention of progressive complications from uncontrolled disease, and studies related to the microbiome. He's also published extensively on the topic of histopathology. So David, thank you so much, and I'll hand over the talk to you from here.
Okay, great. Thank you, Vip. And I want to thank you for including me in this great program, and certainly I'm enjoying the lectures and happy to be with my colleagues. The title I was asked to speak on was Histopathology as a Treatment Target, Aspiration or Reality. I'd like to modify it and say aspiration and reality, because as I think I'm going to convince you, we're close to being there. And I think that if I don't convince you, then certainly Rish Pai in his lecture will. We're going to talk a bit about what we've learned in the field now and where we are. I do have some disclosures that may be relevant because I'll talk a little bit about therapies, but these comments and these slides are all my own. You can go back to the earliest days of people studying ulcerative colitis to appreciate the role of histopathology. This is one of my favorite old studies. It was by Sidney Truelove when he was an assistant physician, and also a colleague of his, Dr. Richards, who was a graduate assistant in pathology. And what they were describing here was the correlation in 1956 between the clinical state, the sigmoidoscopic state, and the histological findings. And what they showed was that even when patients were in clinical remission, they had histological findings of mild to moderate or even severe disease as defined at that time, with different grades of histological severity. And that even when patients had mild to moderate sigmoidoscopic activity, there was a spectrum of histological findings as well. So we've appreciated for many years that the clinical state of the patient with ulcerative colitis or Crohn's disease, as well as the endoscopic appearance and the histological appearance, don't always correlate. So the question when you're doing clinical trials, developing therapies, and in clinical practice is, what should you rely upon, and what do we need to do to raise the bar on how we manage patients? So why is histology of relevance in IBD? Well, that's almost rhetorical, right? It's thought that biopsies will provide more objective evidence of the disease activity. And we also recognize that histologic quiescence, the absence of acute active inflammation, represents a more stable disease control. And through a number of different studies, and I just referenced a couple of them here, the degree of inflammation is associated with clinical endpoints of interest, like time to relapse, or of course, risk of neoplasia. There are a number of different histological scoring systems, and Rish will probably cover this much more thoroughly than I. But the three major systems that we focused on in the modern age and certainly have been focused on in clinical trial development are the Gabos score, the Nancy index, which is from Nancy, France, and the Robarts histopathology score. You can appreciate that each of these scores has a variable degree of descriptors for different categorical assignments. You also might appreciate that none of these scores includes a completely normal mucosa on biopsy, which has been an area of interest for us. But nonetheless, all three of these have some benefits and have been used to various degrees. The one that has been used the most is the Gabos score, and that doesn't mean it's the best. In this great systematic review by Rish that I wanted to quote for you in GI endoscopy a couple of years ago, I want you to just scroll down from top to bottom as chronologically the studies that have looked at clinical outcomes and correlated them to histological outcomes. You can see in the fourth column there the different histologic scores that were used. And if you just scan your eyes over to the last column to the right, you can see what specific clinical outcomes were associated with histological features of interest. And in general, the presence of active histological inflammation at the time of assessment was independently predictive of several different clinical outcomes of interest. Most often, it's clinical relapse. Other times, it's need for steroids. And in some studies, it's even looking at a downstream outcome like needing colectomy or surgery. But obviously, there's a wide heterogeneity of how this has been studied and how people have tried to define the histological activity. Britt Christensen was an advanced fellow here at the University of Chicago for a couple of years. She now runs the IBD Center in Melbourne, Australia, and just 2 weeks ago received her PhD for the work that you see here. So I'm very happy to give her lots of credit and kudos for this. Britt described from our center that about 10% of our patients with chronic ulcerative colitis actually had normalized their histology. There was no evidence of acute or chronic changes that suggested the presence of ulcerative colitis, yet in previous biopsy assessments, they clearly had UC. And most relevant to us in the clinical world and in thinking about trials, normalized patients had a lower likelihood of relapse than those who even had quiescent disease, and certainly better than those with active disease. Now, I'll share with you a number of other analyses that are of interest to you as we go forward here. This particular nice meta-analysis of 13 randomized clinical trials shows you that the odds ratio for remission was higher in those who received therapy and who achieved histological remission compared to other potential interventions and outcomes. Another way to say that is that active therapies that have received regulatory approval for the management of ulcerative colitis in general are associated with both clinical and endoscopic, but also histologic remission. And this has become of great interest. And although it's usually a secondary endpoint or an exploratory endpoint, I'll tell you how it's going to move the field forward. Now, I'm here to share with you a sneak peek at an oral presentation for DDW. Our current advanced fellow, Shintaro Akiyama from Japan, just looked at de-escalation of therapies in patients who had normalized histology. And the interesting thing that he found was that they were stable, but they weren't actually immune from having relapse. In other words, normalized histology in ulcerative colitis, at least in our cohort, did not mean that these patients were cured. Maybe that sounds obvious, but it's important. And we're no different than patients who continued their therapies, suggesting that maybe they're safe to come down on some of their treatments. And there's a lot more we could talk about there. But that's the obvious thing we want to know, is if you get to a deeper level of remission, are you able to use a lower intensity of anti-inflammatory therapy? And of course, that's of great interest to the field. Now in press is also this consensus paper called STRIDE 2. STRIDE 1 was published in 2015. And the STRIDE 2 consensus was from the International Organization for the Study of IBD. And Don Turner, who's the first author here, as well as the other colleagues who contributed, defined short-term targets, intermediate targets, and long-term targets for our treatments in IBD. I want to focus your eyes on the right side here, which is that the consensus statement did address histological healing, but considered it to be not yet a formal target. In other words, we shouldn't be adjusting therapies to achieve histological healing. On the other hand, if you do achieve it, you know that that patient has achieved a stable outcome. For Crohn's disease, the interest was in transmural healing, which of course is a topic for a future conference. So now moving on, let's talk a little bit about how we get to our clinical trials and where we should go. So in order to adapt histopathology as an endpoint in the IBD clinical trials, we certainly need some regulatory guidance. The FDA actually issued draft guidance in 2016, and you can see underlined there in the fine print that they defined mucosal healing as endoscopy in conjunction with histology in order to actually get that label. Otherwise, they give labels of endoscopic improvement. And I actually very much support being more specific in the language that's used. We also, of course, need a clinically validated technique and scoring system with reproducibility of results by different investigators and an understanding whether centralized reading improves the outcomes as well as the reliability of such things, correlation with disease activity, and association with meaningful short-term and long-term outcomes. And although we think of biopsies in Crohn's and colitis as being overall safe, we do need to at least consider that it's still an interventional procedure, and there are, although rare, complications that have to be factored into using this. The other challenges, of course, is recognizing the patchiness of disease activity, not just in Crohn's, but in UC as well, that when we take these biopsies, we're only sampling a small surface area, and therefore we have to understand if extrapolation to the rest of the organ is relevant. It also requires an endoscopist to be thinking about where they should biopsy. The tendency for people is to biopsy when the disease is worse, areas that are less involved, not grabbing the areas that are hemorrhaging or friable. And when the disease is mild, people tend to biopsy the areas that look involved. So there's this bias that goes in both directions. And of course, there are many different people involved when we do this. It's not just the pathologist and the endoscopist. It's people who prepare the samples, and it's a variety of other issues that can be complicated. So we have to think about how many biopsies do we need? Is it sufficient in colitis or Crohn's to biopsy just part of the bowel, or do we need to think about biopsying segments in certain reproducible ways? Where do you biopsy? The picture on the left is actually an ulcerative colitis patient who is on active therapy, and you can see some patchiness endoscopically to the response to therapy. And on the right, of course, is Crohn's disease, and you can appreciate where you might have very different results depending where you grab your tissue. And of course, it's like the old story of the 4 blind people who come up on an elephant and each feels a different part. If you give a pathologist only the biopsies that you take without explaining where they're from, you're going to have different interpretations. And that's a very important point. Now, in our clinical trials, we are finally moving to a point of trying to adopt all this. Ustekinumab is an anti-IL-12/23 monoclonal antibody approved for both Crohn's disease and ulcerative colitis. And in the pivotal trials and the program that led to its approval in ulcerative colitis, which was called UNIFY, Janssen Pharmaceuticals for the first time adopted this combined endoscopic and histologic endpoint. And they called this the histoendoscopic improvement or histoendoscopic mucosal healing. And you can appreciate here that it was possible to do so and that there was benefit to being on a higher dose in maintenance phase, as shown by the light green bar here, which represented that. So this was possible. And very importantly, what we learned from this was also that it predicted outcomes, that it was the people who achieved both histological and endoscopic remission were less likely to have relapse or loss of response and less likely to need steroids over time. So it is associated with this, and very importantly, it was associated with improvements in certain biomarkers, as you can see on the right, like calprotectin and CRP. So can we get from one of these endpoints to a secondary but meaningful biomarker that's been benchmarked to it? More recently, in other emerging therapies, you can see here the anti-P19 therapy, mirikizumab, which is a pure IL-23 inhibitor. What I wanted to show you in this particular phase 2 trial was that histologic remission, as defined in the trial, was better than endoscopic remission. And this is another important point. We don't see correlation either at the same time point or perhaps even in the same amount of time in follow-up of how people heal histologically and how they heal endoscopically. Look at this novel small molecule called filgotinib, which is an anti-JAK1. I want to show you the same thing. Histologic remission in this trial was superior, certainly with drug compared to placebo. And getting back to the question earlier about the liver, you can see there was a placebo histologic remission, but you can also appreciate that it was magnitude greater than endoscopic remission. So is histology better? Does it happen earlier? Or is this just a matter of different definitions and we're confusing ourselves with terminology? I think that remains to be seen, but it's a really important point to understand. Now I'll end with just a couple slides. This is an ongoing trial called Verdict that VIP is leading, in which patients with active ulcerative colitis are actually going to be, or are being randomized to three different groups. One group is where they're doing essentially a treat to target strategy based on symptoms. The second group is treat to target symptoms plus endoscopy with adjustments of therapy. And the third one combines symptoms, endoscopy, and histology. And fully half the patients in this trial are getting treated that way, with the idea that are you going to achieve stable, better outcomes by pushing patients there? I would argue that it's also a feasibility question too. Can you actually do it, and how often can you get there? And lastly, and I'm sure Rish is going to talk more about this, is the international consensus on how to use histopathology in UC clinical trials. So I won't steal his thunder here, but we have some guidance from our pathologists as well as people like VIP in the clinical and clinical trial arena. So in summary, I think that we are moving towards histology, more in ulcerative colitis than Crohn's, but I think in both areas it has very important aspects to what we do. And obviously, there's a lot more work that we need to focus on in order to get there. Thank you very much.
David, wonderful. Thank you so much for such a comprehensive overview of a big topic in that talk. It was a wonderful talk. Thank you. I know we're running about 10 minutes over, and if people can't stay on, you will get a recording of the lecture. But if you can, we're going to have Dr. Pai talk next, and then we're going to have some Q&A. So I'd like to introduce Rish Pai now. Rish received his MD PhD from Case Western School of Medicine in Cleveland. He did his anatomic pathology residency at the University of Chicago and hepatology and GI at the same place. He's professor of laboratory and pathology medicine and consult pathologist at Mayo Clinic in Arizona. He's lectured widely on GI and liver diseases, and he's on the editorial board of the American Journal of Surgical Pathology. Rish has extensively published in this field, and he's really worked extensively with clinical investigators in developing and validating indices in IBD, NASH, and EOE, and has been a wonderful collaborator over many years. Rish, thank you for telling us about how we measure histopathology in IBD clinical trials. So over to you.
Well, thanks so much, Vip, for that kind introduction, and it's really a pleasure to speak to everyone, and thanks to Alimenta for organizing this really important session. So I'm going to be talking to you about measuring IBD or measuring histopathology in IBD trials. And this is the outline that we're going to talk about. I'm going to talk a little bit about scoring systems in UC and Crohn's, and really spend a lot of time talking about how to get this done in clinical trials and some of the issues that we encounter. And we'll spend some time talking about the histologic target that Dr. Rubin so clearly discussed already. So, why are we discussing this? And as David mentioned, the FDA guidance in 2016 really kind of highlighted the importance of histopathology in addition to endoscopy. And there were even some clear guidance about how histopathology should be done. It should be done centrally using standardized methods. Grading scales should also be discussed with the FDA. And this was in 2016. And the real question that I want to ask and kind of address today is really how far have we come since these guidelines were published? So, clinical trials have moved towards more objective measures of inflammation, and endoscopy is still clearly a really key endpoint, of course, in IBD trials. And so we're used to understanding the Mayo endoscopic score and assessing that with a validated index centrally at baseline and then after therapy. So the real question now is, well, how can we do it for histology? How can we take a score? What can we measure at baseline and after treatment? How do we operationalize this? And how do we get to a score that we can trust? So there are numerous indices in ulcerative colitis that Dr. Rubin mentioned. In this Cochrane review, there was 30 indices that we found in UC. And in Crohn's disease, there's also quite a few indices that have been published. And most of them are a variation of the GHAS index. So again, there's a lot of indices out there. How do we hone in on the one to use? And it's really how we assess things like validity, reliability, responsiveness. All of these things are key features that we need to assess for each of these indices to get to a point where they can be used in a clinical trial. And I'm just highlighting the ones in red. These are the ones that have been that have undergone the most extensive validation. And this has been updated to more current date because the Gabos score and the ANSI index and Robarts index have really, I think, undergone fairly extensive validation now. And we know a lot about these the operating properties of these indices that I'll be talking about. So the Gabos score, what is it? Well, it was really developed as a classification scheme rather than an index designed to be responsive. And it measures the whole host of features that you see in UC. And again, it wasn't designed to be responsive, so it includes items like structural change. That it was a useful classification scheme, but responsiveness was kind of not its main goal. These are just, as a pathologist, I have to show you images of IBD slides and just giving you a sense of what it looks like. And it is kind of generally true that the higher the Gabos score, the more severe inflammation you see, and you get to a Gabos 5.4 on the bottom right, which is ulceration, and that's a fragment of ulcerated mucosa there. So the issues with Gabos that I mentioned, it was used as a classification tool to evaluate the mucosal abnormalities in ulcerative colitis, and it wasn't developed to be responsive to therapy. No standardized way to measure improvement, and there was limited data on reproducibility. So in collaboration with VIP and others, we wanted to develop an index specifically designed for clinical trials. And so we used the Goodwill score as kind of the basis because it really was a comprehensive index to evaluate a lot of different features. But we thought we could take those items that were responsive and reliable and develop a new index. And so these are the 4 items that we kind of took from Gabos that are in bold in the right table, which were shown to be responsive by intra-rater ICCs, shown to be reliable by the intra-rater ICCs, either substantial to moderate reliability. We then plotted these different features according to a global assessment of disease activity by a pathologist using the visual analog scale. And we took those features that have a strong relationship and that correlated strongly with the visual analog scale to really derive to take those features that really do represent disease activity. And there were those 4 features that I bolded in the last table, which was chronic inflammation of the lamina propria neutrophils, epithelial neutrophils, and then surface injury or erosions/ulcerations. So this is the Robarts Histopathology Index, 4 features, and you can see how you can get to the RHI points in the far right, and it goes from 0 to 33. So again, this was taking what we know about the histopathology of IBD and really trying to get an index that measures disease activity. And this has been shown in multiple different studies. I just want to highlight that, histology and endoscopy don't always correlate, and this is just real world data, well, actually from a clinical trial, looking at RHI plotted against the Mayo endoscopic score. And you can see that they generally do, correlate. The higher the endoscopy score, the more severe, the higher the RHI. But there's clearly patients with Mayo endoscopic score 0 or 1 that do have histologic inflammation. So again, histology
I think can be a useful adjunct to endoscopy. Because we really want to get at this question, trying to figure out those patients that are truly in disease remission. This is 2 patients. The one in the upper is, but they have a Mayo endoscopic score of 0, or at least to me, the mucosa looks pretty normal. But in the biopsy, you can see active disease with cryptitis. The RHI is 11. In the bottom, the RHI is 0, and the Mayo endoscopic score is 0. So there's really good correlation there. But we really want to use histology to find these patients that are truly in disease remission. Similarly to when we developed the RHI, the Nancy Index was developed based on evaluation of 200 slides from multiple different institutions, and pathologists were involved. And they developed, I think, a really nice score as well, ranging from 0 to 4. And this is kind of the algorithm of how you can score it. And so we really wanted to take it further and say, okay, can we use these scores? Can pathologists really agree on all of these scores? And we took 4 of these scores, including the RHI and Nancy Index, and measured reliability by 4 pathologists. And there was really pretty outstanding reliability here. So I think to David's question, or to the questions that Vip wanted me to address, is can we do this? And I think the answer is yes. We can. That I showed at the beginning, this is the baseline biopsy, and we can now, when I raised the question, can we really score this? And the answer is now, yes, we can. These are the different scores you can get depending on the index used. And the RHI in this would be 26. And then this patient went to this, and we can now measure the disease activity, and the RHI is 1. So I think we're coming to the point where we can quantify this, we can reliably measure it, and it clearly can show a response to therapy. And this is an additional study where we did look at specifically response to therapy in a post hoc analysis of the ozanimod phase 2 trial. Just showing the raw numbers in the table, you can see the placebo response versus active drug, and you can see the RHI and Nancy both did improve. And then a receiver operating curve on the right, showing that all these indices can discriminate between placebo and drug with similar levels. So getting to the issue about how do we measure this, and this is taken from that paper that David referenced in Gastroenterology that we just published, where we convened a panel to talk about all these different issues and where to biopsy, how many biopsies, how to score, et cetera. And these are some of the things that the statements that were appropriate. In the interest of time, I won't go through all of them, but that paper is out there. I think there is one question that I think is going to maybe be a little bit of a challenge operationally, is that there was some consensus that histology should be incorporated at baseline to have some baseline histologic activity, and that could be operationally challenging. In Crohn's disease, this is a recent, this table, it's a busy table, but just wanted to highlight the reference is covered up a little bit, but we just published this in APT, where again, a panel was convened to really some important things that came out from this, including the GHAS score, which is commonly used in Crohn's, may not be the most appropriate index to use, and really a new index is needed for Crohn's disease. So I think in Crohn's, there's still more work to be done. These are some histologic targets. So now that we can measure it, what are the targets? And I've just listed these here. If I won't go through them in great detail, but it does illustrate that certain indices are not cannot be used for certain outcomes. For example, histologic improvement, which is what David talked about in the Stellara trial. The Nancy index cannot be used to assess that, but the Gabos and RHI can. So again, depending on your histologic target, the different index might be better, utilized. And this is just some of the histology of the different targets: normalization versus remission but basal plasmacytosis, and then histologic improvement with just rare neutrophils, which is what the ustekinumab trial measured. So these are some unresolved issues, and I'm glad that David mentioned the verdict trial. I think that's going to be a really key a trial that will really tell us, can we do this? Can we treat to histologic remission, and what that really means? We still need to determine how the bowel heals during therapy. Need to determine the baseline impact or the impact of requiring baseline histology measurements. Is this really possible? Should this be done in clinical trials? And of course, standardizing biopsy protocols, as David mentioned as well. and then standardizing histologic outcome measures as well to compare across studies. So I'll pause there and turn it back to Vip.
Okay, great, wonderful. Thank you, David and Rish, for that session. So we've got to just take a few minutes for some questions. Maybe I'll start because we've got two pathologists on the call. So just tell me a bit about eosinophils. I've always been intrigued by eosinophils in IBD. They didn't make it into some of the new scores, and they're harder to measure. The inter-observer agreement was poor. But what do you think their role is in IBD specifically?
Yeah, I'm sure Mark can answer this as well, but eosinophils are a normal part of the mucosa in the colon. So just their presence, what we're looking for in terms of abnormality is just changing in density, which is something a pathologist has a difficult time with. Are they increased versus not increased? Whereas in neutrophils and some of the other features that we measure, they're not normally seen in the colon, so it's a yes or no kind of thing, and we're used to those kinds of questions. And the number of eosinophils that are abnormal have not really been well defined, and the significance of eosinophils in disease activity and response to therapy is still uncertain in UC, which also makes it a little bit more of a nebulous kind of cell. We really, I think, need to understand more about how eosinophils contribute to UC. And find a better way, probably using quantitative counting or some AI method to measure eosinophils.
Right, I agree with all those comments. And another factor is that we often see eosinophils as a drug effect, and you see more eosinophils in pediatric cases too. So eosinophils are complex, right? And so they may not always be representative of disease activity. In some cases, it seems like areas of neutrophilia have fewer eosinophils. So their role is still a little bit unclear to me.
Great, thank you. David, maybe I could ask you, I think you raised a really important, you put a couple of really important slides. One which stood out to me was the filgotinib study, where he showed at the same time point larger deltas for histological remission compared to endoscopic remission. I think a few trials have shown that. So in your view, does that raise the potential for histology being a more sensitive endpoint? Possibly, could it even replace endoscopy together with symptoms as an endpoint if it's more efficient as an outcome measure? Accepting the evolution of things might be different. You touched on an important point. I just wanted to get your views on that.
Yeah, I think that we need to understand this, but I will say to you that it's entirely possible that knowing that histologic improvement and then histologic remission occurs before endoscopic improvement, you could imagine, if this is validated, using that earlier to know if a therapy is working before we have to use a scope, or even avoiding a scope altogether. One of the unanswered questions right now is how to best sample the mucosa in a way that gives you a reliable answer to what's going on in the rest of the organ. But imagine if we could do this without having to do endoscopy, with a prep and with anesthesia, but rather could pass a brush into the rectum and get some measure that would be more predictive than necessarily doing an endoscopy and looking with the scope. So I think there's some really interesting concepts there that need to be panned out further. I'll just add that I'm also interested in the correlation between calprotectin, which is a marker of activated neutrophils and macrophages. Maybe that's correlating better than we have given it credit for, certainly in ulcerative colitis, to histology rather than to endoscopy, and some of the messiness has been related to that. My last point about this is that the other outcome of interest that's evolving is functional. In other words, rectal compliance and the way the organ actually functions. So maybe endoscopy is not as good as we want it to be once you start treating somebody.
Okay, great. Thanks, David. And maybe I could just ask another question. You touched on this about healing of the colon, and we're always taught that the disease starts left and spreads proximally and it heals in the opposite direction. But you touched on patchiness, and it raises the question of whether sigmoidoscopy sufficiently assesses disease in the whole colon. And what's your sense of that?
Well, we published a paper a couple of years ago in CGH because for years I had noticed that patients with PSC had more histological activity in the right colon than in the left. And of course, we know that they also tend to have less symptoms. And we also know that dysplasia and cancer and PSC-related colitis seems to occur more often on the right. So that's a very specific example. We're doing a sigmoidoscopy is not going to give you a good idea of what's going on in the right colon if we're trying to treat to histological control. And so there's one very specific example. I think, however, that understanding and trying to better study this long-held belief, I would almost say myth, that the bowel heals in one direction or gets inflamed in another is a very important concept. We're biased because it's the rectum that drives symptoms, and it's the rectum that drives people to the doctor and how they get diagnosed. So we don't actually know the real answer to this. I don't know. I'm sure you have some thoughts about it, Vip. What do you think?
No, yeah, I think the same. I think it's a dogma that's been handed down, and I think healing probably is patchy in some way. I do think sigmoidoscopy probably gives the most conservative estimate of efficacy, which is what we want when we're testing therapies. Aside from your example about PSC.
Yeah.
Great, and maybe one very last quick question for Rish and Mark. How many neutrophils are acceptable after you're assessing? Efficacy is less than 5% of the crypts okay, or is that too loose? Should it be no neutrophils? How many neutrophils is okay to see?
Yeah, I'm not a that HEMI endpoint, histologic endoscopic mucosal improvement that allows a little bit of neutrophils, I think is a bit maybe premature. I'm not quite sure that is really, for histology at least, allowing neutrophils to be there really is appropriate. It clearly was associated with some more improved outcomes, but we have to remember that neutrophils have a short half-life, so the fact that you see them means that they were recruited within the past few hours or maybe up to a day. So there's still some active recruitment to neutrophils if you see them. Any neutrophil in the lamina propria or epithelium is abnormal in the colon. So I think it's fine to ponder all these different levels of neutrophils, but I think simply reducing them to zero is really how we should define remission. So maybe as a pathologist, I want purity when maybe it's not possible to achieve sometimes.
I totally agree with Rish on that. As an endpoint, it may be difficult, but normal tissue has essentially zero neutrophils in it. The other point is whether or not we include basal plasmacytosis as well. And obviously, to fully normalize the tissue, you have to get rid of the plasma cells as well in the basal aspect, the majority of them, right?
Great. Well, thank you, everyone. And I think, again, apologies that we've run over time. The recordings will be released in time and any questions will be answered. I want to thank Mark, David, Rish, Stephen for all the time and effort that's gone into today, everyone for joining. And we'll be back in a few months, moving further up the lumen, up to the upper GI tract and talking about EOE. So stay tuned. So again, thank you, everyone, for joining, and enjoy the rest of your days, evenings, mornings, wherever you are. Thank you.